Kylie Swisher, and I am at the USDA ARS research station in Prosser, Washington. And I work with Jim Crosland. And I've been there just over a year looking into psyllid population mapping. And the reason why we became interested in this was because back in 2011 we had ZC in Washington, Oregon, and Idaho in the Northwest for the first time. And so we wanted to ask the question, what psyllids were in that region, what psyllids brought in the Light-Bearer-Bacter. And so we knew from previous research done in other labs, there were two populations of psyllid, the western and the central, in the different geographical regions in the U.S. And so our question was, could we identify the psyllids in the Pacific Northwest during 2011 season? And so instead of doing a lot of DNA sequencing, we went ahead and used a technique called high-resolution melting analysis, or HRM. And this is essentially a tool that allows you to analyze a lot of different psyllid samples without having to sequence everything. And so you can actually see visualize the differences in DNA sequencing by melting profiles. So using this, we saw a couple of really interesting things. In the Pacific Northwest, there were actually two populations of the psyllid. The western population that was previously identified in Baja, Mexico, and California, but also a new northwestern haplotype that we identified for the first time. The other thing that was interesting is that psyllids that were collected in fields hit with ZC in the Pacific Northwest, psyllids from those fields were all of the western haplotype, and we didn't find any northwestern in this region. So this would suggest that the psyllids that came into the region carrying the Light-Bearer-Bacter, wherever they came from, were the western haplotype and were likely from the south in California and migrated their way up into Oregon, Washington, and southern Idaho. Since that time, we've also looked at archive psyllids. We wanted to see if we can get an idea for psyllid dynamics over several different years. We had psyllids in the Pacific Northwest dating back to the late 1990s. See if we can get an idea of what psyllid population has been there this whole time, or when do we see an influx of one population or another. We're also looking at 2012 psyllid collections and seeing if we can monitor the progression of the different psyllid haplotypes over a season, looking at weekly collections.